now I'd like to call upon stage our next speaker. Very proud to call on stage Dr. Krishna Murthy, who was our own fellow from Shankanetralaya, and now he has established himself as a leading researcher and expert in ROP. He works in Prabha Clinic and Vittala Institute of Ophthalmology in Bangalore. Dr. Krishna Murthy will be speaking to us on urinary biomarkers for retinopathy of prematurity.
At the outset, I'd like to thank Dr. Pramod Bende, Pradeep, Chetan, and the entire organizing team here in SN for putting together this wonderful program and also making me a part of this. Proud to be back here. Of course, it all started from here as a research fellow. Binocular indirect ophthalmoscopy is still the gold standard for screening for retinopathy of prematurity. But we understand that there's a great mismatch in the number of babies who need to be screened to the number of available ophthalmologists to screen them. With the advent of telescreening and teleophthalmology, this mismatch has been reduced to a large extent, no doubt. And you'll hear more success stories in the next session. But we have to agree that there's still a large number of babies who do not have access to ROP screening. So the goal here was to develop a cost-effective and a non-invasive pre-diagnostic ELISA screening panel for the diagnosis of retinopathy of prematurity in premature babies from their urine samples. The objective here is to identify all those significantly dysregulated proteins in the urine using an untargeted, unbiased, high-resolution mass spectrometry-based quantitative proteome profiling from the urine samples of ROP and control babies. We also wanted to look at developing an antibody-based ELISA methods to predict the ROP in premature babies for these shortlisted candidates from the dysregulated protein list. Finally, we need to validate these candidate protein biomarkers of interest on a statistically significant cohort of patients using our ELISA methods. So the workflow of our experiment. So my talk here is largely based on our own experiences in working with urine of ROP babies and looking for biomarker discovery. So the urine samples consisted of samples from babies who were undergoing screening for retinopathy of prematurity. These were babies who were less than 34 weeks of gestational age or less than 2000 grams birth weight. These urine samples were then subjected to untargeted proteomics, which was based on high-resolution mass spectrometry, and we used the labeling technique of tandem mass tags. This was the discovery part of the experiment, which led to the identification of a significantly large number of dysregulated proteins. These are upregulated or downregulated proteins. From this list, a short list was created, which was based on fold chain. That is basically how much more concentration was found in the diseased urine samples compared to the controls. Also a bioinformatic network analysis, bioinfo analysis, and a good literature survey to see which candidate would fit into your biomarker panel. And finally, develop and validate these shortlisted proteins on ELISA-based assay panel. The boxes in yellow are work which has already been done. The box in gray is work which is ongoing. I'll be presenting part of the work which is here and of course all the work done there. The final outcome we are looking at is, can we predict ROP from the urine samples of these premature babies? So the workflow for the quantitative proteomic work, this is the discovery experiment, was such. We took 4 urine samples from babies with retinopathy of prematurity and 3 urine samples from babies who did not want to develop retinopathy of prematurity, which acted as the controls. The urine was concentrated, the protein was estimated, and it was digested with trypsin. After digestion, the samples were tagged with tandem mass tags. These are basically isobaric tags. And once you do that, we subjected the samples were pooled and subjected to basic reverse phase liquid chromatography, followed by a CMS-MS analysis. So this threw out a list of those proteins which are differentially expressed between the ROP samples and the controls. The reason for the TMT labeling is that once all these samples are labeled with a particular mass tag, then once the mass spec picks up the protein, it can exactly say from which sample this protein has come. Hence, it can quantify it in each of these samples separately. We found about 2000 proteins identified, of which 129 proteins were significantly dysregulated. We consider a cutoff of 1.5 times. Then these dysregulated proteins were passed through a network analysis, string database network analysis. So basically, you're trying to see all these dysregulated proteins which we have found, many of them here. We pass them through different biological processes, which could include blood vessel development, circulatory system development, angiogenesis, vascular development, or vessel morphogenesis. This is a bioinformatic tool. And we did find that many of our proteins, dysregulated proteins, were largely present in the network biological processes related to angiogenesis and vascularization, which was by itself a partial validation of our study. Next was to look at the candidates for shortlist this list into a smaller list for ELISA validation. We chose 6 proteins: cystatin 6, fibronectin, vitronectin, and cadherin 2. The reasons for choosing these 4 were: one, their abundance in the urine, whether you could find them at all or not. The second one was the fold chain. How much more was they increased in the disease samples compared to your normal samples? You can see here that cystatin is almost 5.5 times more than in controls. Fibronectin was 3 times more abundant in the disease than in controls. Vitronectin was 4 times more abundant, whereas cadherin was about 5 times more abundant. We also subjected these, saw what they did in these biological processes, which you saw in the previous slide, which was the string database based network analysis, and lastly the literature survey. We have worked so far with 2 proteins, which is fibronectin and cystatin 6. The workflow for the validation process, ELISA validation process, was such: the urine samples were again divided into those with ROP and those without ROP. We found the concentration of, we understand here that urine is not an isotonic solution. So it is, it is going to be influenced by the fluid intake of the baby previously and things like that. So absolute value calculation of these proteins is not going to be of much use. So you need to find some way to find whether it is actually raised or not. So here what we did was we found the concentration of creatinine, and we also found the concentration of our selected protein. And then we estimated this ratio between creatinine to the protein of interest. It was done so in both controls as well as in the subjects of ROP. And then we compared the ratios, because if there is a dilution factor, then this ratio should eliminate the dilution factor. And then those group of proteins with high concentrations in the ROP urine samples compared to the controls formed your biomarker panel. The reason why creatinine was used is because creatinine is largely not influenced as it is passes out through the urine. So basically, it's metabolically not affected as it passes out through the urine. In fact, creatinine was used to estimate GFR before the newer molecules which have come in there. So that's the reason why the comparison with creatinine was done. The results for cystatin and fibronectin were significant, very significant. In fact, cystatin was more than 2.7 times increased, and fibronectin was almost double of what the controls was. And this is a whiskered plot, the same ELISA results which have been plotted on a whiskered plot, and you can see that it more or less matches with what we had seen with the mass spec. So to conclude, the dysregulation of the selected candidates was quite significant, and which for the number of samples that we have analyzed so far, with cystatin showing almost 2.7 times increased abundance and fibronectin showing almost 2 times increased abundance. We are in the process of testing a larger number of cohort samples with 4 different candidates, which I just shared with you. The quantitative results which obtained from this combination of these markers may direct us in establishing a scoring system using again bioinformatic tools, using an artificial neural network system. This is what we would like to work on next. This system of analysis may help us in predicting the ROP in preterm babies much earlier, or at least without the formal screening that we already do. I would definitely like to acknowledge our neonatologists, Dr. Niranjan, Dr. Pratik, and Dr. Naveen Benkappa. Vinod and Arun Kumar have been helping me with the ELISA results, and Dr. Akhilesh Pandey and his team at Institute for Bioinformatics have done the mass spec work, and and the entire tele-ROP team of our own institute to visit corners of Karnataka and screening many babies and treating them as well. Thank you.
Thank you, Dr. Krishna. Excellent talk.